Good morning. Thank you for the introduction. I'm so excited to be here speaking to you all today, representing Ultragenyx. Up until a year ago, I used to be part of the academic panels for patient advocacy groups, and now I'm part of industry. So today we'll be talking about some updates from Ultragenyx's UX055 program. And as explained, I'm the global clinical development lead, bringing forward our program from preclinical to the clinical stages. So we'll give you an update today. We'll talk a little bit about Ultragenyx. It's important to know how Ultragenyx approaches rare disease drug development and explain to you how patient engagement is a critical part of our clinical development program. And we want to learn from our patients the truth about a disease. We'll provide a brief status update about UX055, our preclinical CDD program, and then explain how the CDD community is helping to advance our program towards clinic. We want to have some discussion and understand any questions you may have for us. So who we are and what we stand for. So Ultragenyx was founded in 2010 by Dr. Emil Kakkis, and his goal was to create a steady pipeline of new and effective therapies for the treatment of rare and ultra rare diseases. I personally met Emil about in 2011 as I was an up-and-coming clinical researcher at NYU. And I've seen this company grow from a group of 12 people up to over 1,000. And he worked with me over the years as I conducted research in my rare disease patients. I ran the undiagnosed disease program and managed several hundred patients with rare diseases over the past 10 years. So this company is headquartered in Novato, California, but we have offices all over in the Bay Area, in Massachusetts. We have global presence in Europe, Latin America, and a presence in Canada. We're dedicated to deep and meaningful engagements with the rare disease community to fully understand and address their needs. So what are our guiding principles for rare disease drug development? What's important to know is that we learn and adapt. We are seeking the truth from patients and caregivers. We think and actively engage patients into all stages of development, even when we're just starting out and looking to develop a proof of concept in the Petri dish, in small animal models. We start talking to patients right away. We invent new ways when needed. So we'll invest in the early stage development, and we collaborate with academic institutions and others. It's important for us that we want to learn and adapt and shape the best way forward to design, to delineate endpoints, to understand how we can benefit the overall disease through information sharing. We are committed to sharing our learnings with the greater community, and that's involved at all stages through regular updates like these. We want to provide multiple ways to win. As you can see, it's very difficult, as you heard this morning, to get a drug approved, and there are very few that are approved in the space for gene therapy, and there's very few for CDD. So we want to leverage multiple paths that we will look simultaneously at and try to innovate and collaborate with others. And that will be highlighted as we go through this presentation today. So successful treatment of patients. I know this as an academic physician and a provider for patients with rare diseases. We need to learn from you directly, from the caregivers, from patients over time. What is interesting, and I think I abide by the same rule prior to joining Ultragenyx, is that we know that rare diseases often have one major well-accepted fact, something that we think is true about the disease, and actually it's wrong. So what do I mean by that? Well, there are certain things that we learn or we're taught in school, in medical school, about a disease state. And then as we get into practice and talk to our patients, we realize that actually this is not a true fact about this. My personal experience is, for example, in Morquio disease, where we thought it was only a bone disease, MPS IVA. But as I was talking to my patients during the pivotal interventional trial for enzyme therapy, I realized that what impacted patients just as much as their bone disease were their respiratory problems and other problems from the neurologic aspects of this typically non-neuronopathic disease. And they were improved with enzyme. So we learned that in real time. Another example is visual impairment, which is kind of relatable to CDD. in that in Canavan disease, a disease I was studying since I was a fellow, we historically thought that the nerve damage was primarily in the eye. But in fact, through natural history studies and working with others around the world, we realized it was actually cortical visual impairment. The problem was in the brain, not in the actual eye itself. And how did this inform us? Well, when we went forward to start developing a gene therapy trial, we had to incorporate this knowledge and not look at the eye, but look at the brain for this dysfunction to see if it's going to be amenable to change. So this is how we learned directly from our patients. This was in real time, learning how a patient is expressing or the burden of disease on that patient, and then understanding the response to therapy. And that's how we can unlock successful treatment of a disease. So our approach to patient-focused drug development, there's 4 stages. When we're in the preclinical and acquisition stage, we then move into early stage, late stage, and commercial, where it's approved and available to the entire population. But at each stage, we aim to address certain questions from the individual and the family unit. What is it like to live with this disease, not just for the patient, but for the entire family? What are the treatment goals for you and your family that guide decisions about management of the disease? And what is important to the family about the current treatments that are going on, the pros and cons? What do you see as a target? We heard Dr. Demera say that we're not just looking for symptomatic control. We want to get to the crux of the problem. So what is a successful treatment? What does it look like for you, the parent? What does it look like for your child? And how do we address these unmet needs? Then we start looking at communities and systems of care. What are the support systems out there? What is the care right now? What can you get from healthcare providers, therapists, and who is at risk for this disease? So we learn about where these patients are being seen and how they're being managed. So from all of that, then that helps us inform as we develop a program for this disease. So what do we most want to know, and what sources and methods of investigation are most appropriate? All right, so this is a very busy slide, but this is where we are at in our stage of development. That was nicely presented this morning by Anna. So we are at something called stage 0 in our company, and that doesn't sound very exciting, but it is actually very exciting for me, because that's where we start engaging further in clinical programs to start up. Now, I did say that we engaged the community all the way back at stage -2, when we're just looking at treatment effects in vitro. And then stage -1 is starting to look at mouse models or actual models of the disease in animals. So stage 0 is this gray area a little bit. So to date, I'm going to summarize. These have been presented at various forums over the past couple of years, and we'll have a further update in July. So we started in our non-clinical area, looking at the pharmacology and efficacy in a male mouse model and female mouse models. We're looking at how does our drug get delivered? Where does it get delivered? In larger non-human primates, so a larger animal model. Are we getting the gene therapy where we need it to go? And then looking at that biodistribution and the toxicology, so the safety of it in these well-controlled studies that are priming us or getting us ready for the clinic. So what is interesting being on this side of things in industry as opposed to in the academic world is now I'm understanding more about the manufacturing part of this. That could be a key block to getting to the clinic. So we have a proof of concept. We understand that our gene therapy is carrying the appropriate gene and hopefully delivering it to the neurons and getting it all over the brain. But now we need to look at that drug and make sure that we can administer it in larger amounts. So as we scale up, we make sure that there's no impurities and that we are producing a good clinical product that will go into humans in the future. So that we're at this stage, we're starting to manufacture. So this stage 0 is where we engage manufacturing and starting to administer, make that drug so we can give it to the large animals and start and prepare for human consumption. In the clinic, so this is my role, and I work with a wonderful team of different backgrounds and areas of expertise, ranging from patient advocates to biostatisticians to people who are experts in endpoint development. And we consult with caregivers, patient groups, and other clinicians to start thinking about the study protocol. And this is where we're at. So we want to understand the diagnostic journey, the burden of the illness, again, the ideal treatment that you envision for your child, barriers to the treatment, and then getting a sense of your gene therapy perspective. What do you know about gene therapy? What are your expectations? And what are your concerns? Are you interested in gene therapy for your child? We start to think about selecting clinical assessments, and we leverage our colleagues in academia and other areas who are looking very closely at the various aspects of CDD, whether it's vision or motor or language or seizures, to understand what can we measure over time in a trial. And we want to start to develop dose administration procedures. The brain has this tight junction called the blood-brain barrier. How do we develop and get this gene therapy to where we want it to go, which is the brain, to the entire brain? What are the strategies? What are the methods to get that across? And that's what we're thinking about now. And then we get feedback on how do we recruit. So we've been conducting literature reviews, retrospective chart reviews. We're initiating a biomarker development, which I'll mention in the next slide, and supporting our academic colleagues in the U01 NIH grant, and also with the Lulu Foundation's Candid study. We're an active participant and supporter. So with all of this going on, there's another piece. I talked to you about the non-clinical, the manufacturing, the clinical, and we could have enough drug and we could be ready for clinic, but we cannot dose patients until we have the go-ahead from health authorities. So this pre-IND stage is where we're getting ready to say to the healthcare authorities, listen, we're ready to go. What do we need to do? What do we have to prove to you to say that it's safe to go ahead with a first-in-human study? So this is called the pre-IND. We get scientific advice from the health authorities, and we start into the IND development phase, which is investigative new device, new drug application and approval. And so all of that has to go on simultaneously while we are honing in our preclinical and our clinical strategies. So to date, we have participated, as I said, in All Stripes study where members of the CDD community allowed us anonymously to work with All Stripes to curate information about your child's disease and how you've interacted with the healthcare system through medical records. And we learned a lot about the disease, and we confirmed what we do know. We also confirmed what we need to still find out prospectively. The biomarker study is an endeavor that we are moving towards, and I'll have an update in July for you all. But what is a biomarker? So biomarkers could be anything. It could be blood samples, it could be CSF, it could be electrophysiology, it could be imaging. There are any indicators, silent indicators of CDD disease symptoms that hopefully we could measure and then see a response when we're trying to test out a new therapy. So our biomarker study initiative is a very small, focused, one-time blood collection for a small subset of patients, just to start this exploration, to start looking in the blood for markers of CDD. And so we have a whole section in our company dedicated to biomarker development, and we're initiating that. And of course, once we have some information, we are going to share it back with the entire community so others can benefit from this. We are continuing our patient-focused research. We had an initial insights meeting with caregivers from the US and Canada. And that small group session was so valuable as we listened to your child's diagnostic journey, understand the potential treatment, the treatments that are out there, and how you envision and what are your perspectives on gene therapy. We went on then to have one-on-one interviews with caregivers. Every time I speak to a parent of a child with a rare disease like CDD, I learn something. I learn something and I incorporate it and I feed it back to our team to see, are we addressing it? Are we incorporating our information? Is it valid? Our second insights meeting will be conducted in both US and ex-US sites to continue this knowledge gathering. And this will all help shape our clinical trial design and process and understand what we need to put into a protocol for future first-in-human treatment. And so all of this is going on in the background as we're preparing towards clinic. So so again, we're interacting with caregivers and advocates. We're starting to learn global advocacy groups and fill in the knowledge gaps. How does CDD get treated in the UK versus the US versus Europe versus Japan? These are all important to understand the different cultural aspects and perspectives of CDD. We're working with key opinion leaders. These are expert physicians and healthcare providers, not just in CDD. So we're engaging with all of the academics, many of the academics involved in CDD research, but we're also engaging with people that are experts in gene therapy. I had the opportunity to conduct gene therapy and help with gene therapy design as an academic. And so now we're working with others who have specific experience in CNS-directed gene therapy. And we want to make sure that we understand and execute our dose administration appropriately. We also work with researchers. So I said it a few times with the Lulu Foundation's Candid Study. We're one of 7 companies supporting them. I sit in on meetings and help guide and answer questions or provide input. And for the UL1 grant, there's 4 of us that sit on the external advisory committee. So as stated, we will have some exciting updates and information in another month. That'll be a webinar hosted by FCR on Wednesday, July 20th from 4 to 5:15 p.m. Pacific time. I'll be presenting again with hopefully some new information and to answer your questions. So please look out for that event. Thank you so much for your time today, and I'll take questions.